Hi, my name is Roshni, and this is my final project for BIOL 105L. In this presentation, I'll be discussing the commonly used technique ELISA, which stands for Enzyme-Linked Immunoabsorbent Assay. This technique has widespread application in a variety of scientific environments, such as in a laboratory, hospital, or in households, such as through pregnancy tests. This presentation will discuss the science behind ELISAs and their applications in the real world. In order to communicate the mechanisms behind this assay, it is important that some basic information about the materials and systems involved in this technique are conveyed. Antigens. Antigens are any pathogen that has the potential to cause harm to the body. Antigens could potentially be bacteria, viruses, parasites, or even malfunctions facilitated by your own body, such as cancer. Antigens are targeted by the immune system to be identified and shut down through a variety of defense mechanisms that serve to protect us. Antibodies. Our immune system is a defense mechanism against dangerous pathogens and external factors that could be detrimental to our health. Immune cells can generate substances called antibodies, which are proteins specific to the pathogen that the immune system is targeting. Antibodies contribute to our immune system in primarily two distinct ways. First of all, they can bind to markers on the surface of antigens called epitopes to mark the antigen for destruction. This allows cytotoxic T cells to identify these marked pathogens and destroy them by releasing chemicals. A second function of antibodies in the immune system is to bind to epitopes and prevent the antigen from entering the cell, allowing for a temporary stop on any further damage. Antibodies are especially useful because they can be synthetically harvested and used to our advantage in therapeutic treatments or engineered assays. Enzymes. Enzymes are proteins that facilitate chemical reactions in the body by acting as a biological catalyst. They act on substances called substrates, which are usually enzyme-specific. Enzymes have active sites where substrates bind and the chemical change is facilitated. Now that we've gone over the scientific background, we can get into the assay being highlighted. ELISAs are a type of assay that utilize antibodies or antigens to measure the quantity or presence of secreted proteins of interest in a sample. There are subcategories of ELISAs that can be utilized in the medicinal and research world. Here, we'll briefly go over some of the differences between the varieties. Starting off with direct ELISAs. Direct ELISAs constitute two parts: a microtiter plate coated with an immobilized antigen, and the antibody with an attached enzyme. When a substrate is added to the mixture, it induces a color change when the enzyme interacts with the added substrate. The color change can then be measured and quantified to determine the concentration of the antigen in the sample. Indirect ELISAs also use a microtiter plate, except in this case it's coated with an antigen instead of an antibody. The objective of this assay is to measure the concentration of antibodies instead of antigen. The mechanism is similar. A sample is added, a secondary antibody with this attached enzyme is added, and the color change is measured once the substrate mixture is added. Sandwich ELISAs are a little different in that they use a microtiter plate that is coated with antibodies, and this allows the substance of interest to bind. If the measured protein comes from one specific species, the antibody is usually sourced from an organism that is not the same species. Once the substance binds to the antibody, a secondary antibody with the enzyme attached to the constant region is added to the microtiter plate. The variable region of the secondary antibody binds to the antigen, and the substrate is added to the mix as well. The enzyme attached to the secondary antibody acts on the substrate, which allows for the color change, and which is then measured. Competitive ELISAs are the most complex. A control antigen is added to a plate, which is covered and buffered. A blocking agent is added containing a protein to block off free sites in the microtiter plate. Once the sample is added, a secondary detection antibody is added, which allows for the quantification. So, there are many applications of ELISAs. They're used widely throughout the medicinal world for diagnosis of many diseases or illnesses. For example, ELISAs can be used to diagnose HIV, Lyme disease, syphilis, squamous cell carcinomas, just to name a few. What makes them especially useful is the compatibility of these diagnostic tests and inexpensive reagents that constitute the test. ELISA diagnostic tests do not simply exist in hospitals or doctor's office, but can also be purchased at local drug or convenience stores as well. For example, one of the most ubiquitous examples are lateral flow ELISAs that come in the form of pregnancy tests. These tests are not quantitative nor fully dependable, but allow individuals to have easy access to results before getting confirmation from a blood test or from their doctor, in the case of pregnancy tests. In conclusion, ELISAs are really useful because they are very sensitive and versatile. Connecting it back to the lab I researched, in the Berlot lab, a direct ELISA was used to quantify the amount of VEGF protein levels in a subclass of retinal cells. VEGF proteins are a type of cytokine that has pro-angiogenic characteristics, the products of genes induced by HIF-related transcription factors. They used the mouse VEGF quantikine ELISA kit along with an acid assay, which is another colorimetric assay, to determine the total protein amount in the sample.